So now it's my pleasure to introduce to you the last talk and speaker. For those who don't know, Stefan Fröhling. Stefan Fröhling heads the division of Translational Medical Oncology at the DKFZ here in Heidelberg. He is also the managing director of the NCT Heidelberg since 2020. He studied medicine in Heidelberg and Marburg and got his medical training in Heidelberg and Ulm. He spent 4 years as a postdoctoral researcher in hematology at the Brigham and Women's Hospital at Harvard Medical School and was later group leader, research group leader in the University of Ulm, also in the field of hematology and oncology. That's one of his research focuses, sarcoma, bone and bone tumor research, the biological part, and also some forms of acute leukemia. But his main focus, or his other focus, why we have him here today, is all his research centered around the development of tools for the molecular characterization of individual tumors and feeding that back to individual patients. And that serves research at the one hand, but also serves the individual patients. And this program is called the MASTER program. We use it as clinicians or oncologists a lot for our patients, but it also generated a lot of research results that are beneficial. So a very translational program, and he will report and explain where he is at with this program at the moment. Welcome, Stefan.
Yeah, super. Thank you very much, Eva, for the introduction, and also thanks to the organizers for inviting me for the opportunity to participate in this fantastic symposium. So I listened to the second part, and I thought it was very interesting. I'm glad to be part of it. So as Eva mentioned, I would like to talk about the clinical value of very comprehensive molecular profiling, i.e. genome and RNA sequencing in the clinical care of patients with solid tumors. And I would also like to explain how we have organized this at our center and also across different institutions in Germany. And as you will see from my talk, this will be clinically oriented. I'm really not an expert in data sharing. Therefore, I won't talk about it too much, but I can perhaps in the end explain some of the problems we are facing in our own program. So I've chosen to illustrate the clinical value of broad molecular analyses using a particular group of tumors that Eva already alluded to, and these are the soft tissue sarcomas. Soft tissue sarcomas are a very heterogeneous group of rare malignancies that can occur in virtually any body region and that are derived from the tissues that are indicated here on this slide. And clinically, a problem is that progress in the drug treatment of soft tissue sarcomas is hampered and has been inhibited by a lack of adequately sized clinical trials, which also often fail to account for the heterogeneity of these diseases. And there are only few clinically validated molecular drug targets when you compare the sarcomas to other more intensively studied cancers, such as epithelial cancers. And despite this really unsatisfactory situation, I'd like to recall, and this is as a ray of hope, so to speak, that although soft tissue sarcomas are often challenging to treat, and we have comparatively few drug therapy options available, they also represent a group of diseases in which several precision oncology milestones have been achieved. And I just wanted to list one or two of them. So this is, of course, the case for the targeted blockade of the mutant KIT receptor in patients with GIST, gastrointestinal stromal tumor. But this is also true for more complex alterations, and this is exemplified here by the discovery of pharmacologically targetable rearrangements of another receptor tyrosine kinase, ALK, in patients with a rare disease called inflammatory myofibroblastic tumor. And this slide just summarizes one of the first case studies published already in 2010 that reported on the therapeutic efficacy of crizotinib, which is a combined ALK and MET inhibitor in this disease, and whose value as a therapeutic target could also be demonstrated in a multi-center phase 2 trial conducted by the EORTC, which found a perfect disease control rate of 100% among patients with inflammatory myofibroblastic tumors that harbor an ALK rearrangement. And another particularly striking recent example are rearrangements of another receptor tyrosine kinase, the NTRK receptors, which, as in many other entities, are associated with remarkable sensitivity to NTRK inhibitors, such as larotrectinib, this is shown on this diagram, or entrectinib in patients with different soft tissue sarcoma subtypes. And so based on these and many other examples, we think that there is no doubt that molecular genetic analyses are already of considerable importance for the treatment of patients with soft tissue sarcomas. However, from a clinical perspective, the question arises to what extent such analyses, which, as you know, can range from looking at single mutational hotspots, shown on the right, to sequencing entire genomes, shown on the left, should be performed. And this diagram illustrates how we have organized molecular stratification at our center, at the National Center for Tumor Diseases in Heidelberg. And on the one hand, and this is the green box, there is routine molecular diagnostics, which primarily involves gene panel sequencing, large gene panels, and targeted gene expression profiling. And on the other hand, this is the blue box. We run several specialized programs that systematically use whole genome and RNA sequencing in specific patient populations to guide clinical action and also to develop novel concepts for molecularly informed interventional clinical trials. And because of its importance for patients with soft tissue sarcomas, I would like to describe the MASTER program, which focuses on young adults with advanced cancers across entities, so patients who are not older than 50 years of age, as well as on patients with advanced rare cancers across age groups. And within this program's framework, the collaboration of 11 comprehensive cancer centers in Germany with their respective catchment areas has resulted in a really close-knit precision oncology network that now comprises more than 100 partners. And it also represents the entire spectrum of cancer care, from university hospitals, comprehensive cancer centers, such as our own center, to oncologists in private practice. So we think that program is truly also a community effort. And since the start of the program at NCT Heidelberg in 2013, the levels of patient characterization have increased and currently include information on the exome or genome. This depends a little bit on the site where the patient is treated, transcriptome and methylome of each tumor, as well as cancer-relevant germline alterations. And I'll come back to that in a second. And using a standardized workflow for recruitment and consenting of patients, sample processing, molecular profiling, and bioinformatic analysis, we have now analyzed more than 3,000 patients at the time of the last interim evaluation. Certainly small numbers when you compare us to MSK Impact, but I guess that the unique thing about the MASTER program is that we use genome and RNA sequencing, and that this is a Germany-wide effort. And the patients that we enroll represent a fairly broad spectrum of entities, of which approximately 75% are rare cancers. And currently we use 468 single gene and 6 composite or compound biomarkers to make treatment recommendations and assign each case to one of 7 different so-called intervention baskets based on the cellular processes or pathways that are disrupted by the respective molecular alterations. And if you look more closely at this distribution, at the pie chart, you will see that the soft tissue sarcomas account for the largest proportion of our cohort, which we think primarily reflects an significant unmet medical need in these patients, who are often younger patients and have only maybe 2 or 3 useful treatment lines available. And the following slides show examples of how the results of molecular profiling, the way we use it, can influence the clinical management of individual soft tissue sarcoma patients. So first, there is a diagnostic aspect, because genome and RNA sequencing result in a re-evaluation of the clinical diagnosis in approximately 5 to 10% of sarcoma patients, which is of course always done in close consultation with colleagues from pathology and may sometimes have immediate therapeutic consequences. In addition, we also observed that some undifferentiated carcinomas, or this was the clinical diagnosis, can be classified as sarcomas based on the genetic data. So we think broad molecular analysis clearly has relevant diagnostic value in the field of soft tissue sarcomas. And secondly, systematic analysis of germline alterations revealed, and this came as a surprise to us, that approximately 10% of our patients have clearly pathogenic variants in established tumor predisposition genes, 75% of which were not known to the affected family at the time of analysis, and which sometimes provide targets for molecularly targeted therapies such as PARP inhibition in patients with inactivating BRCA1 or BRCA2 or PALB2 mutations. And in addition, approximately 5% of patients are carriers of autosomal recessive disorders. And I should mention that the whole germline program is the work of Evelyn Schröck and her team at our partner site at the NCT in Dresden. And thirdly, and that's perhaps the most important aspect, comprehensive molecular tumor profiles often provide us with starting points for individualized sarcoma therapies. And this is just an individual case, and I'll show some of them. This is the case of a young woman with a very rare disease, metastatic pulmonary epithelioid hemangioendothelioma, and no response to 2 lines of standard systemic therapy. And her tumor was found by exome sequencing at that time to harbor a focal amplification on the long arm of chromosome 4 that affected, among other genes, the PDGF receptor alpha gene, and was associated with very high expression of that gene, as well as its cognate ligand. And these findings provided a molecular rationale for treatment with a multi-tyrosine kinase inhibitor called pazopanib, which resulted in a partial remission that lasted for approximately 6 months. And while this patient is an interesting individual case, an important question is, of course, whether new starting points for molecular stratification of larger patient groups can also be found. And that this may be the case, and we have now several examples, is illustrated by this case. This was a young woman with refractory retroperitoneal leiomyosarcoma and amplification of the FGFR1 locus on the short arm of chromosome 8, who benefited from pharmacologic FGFR blockade in the context of 2 clinical trials for more than 1 year. And based on this clinical observation, an individual case, we have determined through analysis of 3 retrospective patient cohorts from Munich, from Heidelberg, and the TCGA dataset, that alterations of FGFR1 occur at low frequencies in at least 7 different soft tissue sarcoma subtypes. And we observed in fairly simple laboratory experiments that the sensitivity of soft tissue sarcoma cell lines to FGFR inhibitors that are in clinical development is primarily dictated by aberrant FGFR1 expression and sufficient inhibition of the RAF-MEK-ERK pathway. And another more recent example is the discovery of an activating mutation in the ERBB2 receptor tyrosine kinase in a patient with a very rare type of peripheral nerve sheath tumor called neurofibroma schwannoma, hybrid nerve sheath tumors. She's being treated at Frankfurt University, and she has been responding to small molecule ERBB2 inhibition for more than 5 years now. And based on this observation, we took advantage of the German Cancer Consortium network, the DKTK, because we were able to analyze 18 additional cases collected through that network and identified or discovered that ERBB2 mutations are in fact recurrent events in a specific subtype of this entity, specifically in hybrid nerve sheath tumors that are associated with sporadic schwannomatosis. And we also observed, and this was a collaboration with the neuropathology department here in Heidelberg, that these tumors are part of a distinct subgroup of peripheral nerve sheath tumors defined by a very characteristic DNA methylation profiling. So we think this discovery is therapeutically useful, but does also have diagnostic value. And the assumption that other pathways besides receptor tyrosine kinase signaling may also have importance as therapeutic targets in patients with soft tissue sarcomas is supported by this case. This is a woman in her 50s with chemorefractory metastatic uterine leiomyosarcoma. And since we identified a biallelic TSC1 loss in this tumor, the patient was started on the mTOR inhibitor everolimus, and this resulted in a very good partial response after 3 months. Again, an individual case, but this result is also consistent with our previous observation in the context of a survey of the genomic and transcriptomic landscape of leiomyosarcomas, that alterations potentially associated with a permanent activation of the PI3 kinase AKT mTOR signaling axis are present in more than half of these tumors. So I think this observation illustrates the value of patient cohorts that we are building in precision oncology programs such as MASTER. And I think that's particularly helpful in the context of rare diseases where we often have no clue how to treat them. And as alluded to earlier, an important aspect is also that our comprehensive molecular profiling approach, so as I mentioned, genome and RNA sequencing, allows us to capture compound biomarkers that comprise multiple different components. For example, whole exome or genome sequencing identifies genomic imprints of defective DNA repair via homologous recombination in nearly all patients with advanced stage Leiomyosarcoma, and also in nearly all patients with advanced stage Chordoma, another ultra-rare disease that we see quite frequently in the MASTER program. And this suggests that the concept of BRCA-ness as a therapeutic liability might extend beyond the common epithelial cancers such as breast, ovarian, prostate, and pancreatic cancer. And the potential clinical value of this discovery is illustrated by another individual case. This is a 57-year-old Chordoma patient who showed an exceptionally strong BRCA-ness phenotype and responded to pharmacologic PARP inhibition, so monotherapy with olaparib for a period of 10 months. In this case, also illustrated to us that sequential genetic analyses within a precision oncology program before and after targeted treatment can be quite informative, because whole genome sequencing of a repeat biopsy from a progressive region of the tumor that we took when he showed acquired resistance revealed a newly gained missense mutation in PARP1 itself that is predicted to disrupt the autoinhibitory so-called helical domain and stabilizes the active conformation of PARP1. And we think this in a way validates PARP1 as a therapeutic target in this particular tumor. And so now I talked about several individual cases, but the overall implications of this stratification approach are nicely illustrated by the first 1,310 enrolled patients for whom we have comprehensive and manually curated clinical information and sufficient follow-up available. And this was recently published. And we made evidence-based treatment recommendations in more than 85% of these patients. And these recommendations were implemented in 32% of the cases, of which 55% had an objective response or stabilization of their disease, including patients, and this is shown in the diagram on the left, who had received recommendations with a low level of evidence. In addition, we observed that the molecular data helped us to clarify the clinical diagnosis in approximately 5% of cases. I mentioned that before. And I also mentioned that systematic analysis of germline alterations showed that approximately 15% of patients have variants in known tumor predisposition genes or are carriers for an autosomal recessive disease. So these are data on response, and these are the data on progression-free survival. So the proportion of patients in whom the recommended treatments were associated with the prolongation of the progression-free interval by at least 30% compared to the last systemic treatment before molecular analysis was 36%. And this included most soft tissue sarcoma patients. And these calculations were based on cases representing a total of 20 different anatomically or histologically defined disease categories, and a progression-free survival ratio of greater than 1.3 in more than 30% of patients was found in 16 of these subcohorts. And if we use a stricter threshold, a progression-free survival ratio greater than 1.5 in more than 30% of patients was detectable in 12 subcohorts. So we think these results are encouraging, given that this is an ultra-high-risk patient cohort, but these results also teach us which patient subsets do not benefit from such an approach. Just to mention one example, bone sarcomas are a tough nut to crack. And the ultimate goal of MASTER is, of course, to move away from assessing the therapeutic activities activity of these targeted interventions on an individual case basis. And towards this goal, we are working on a continuously growing portfolio of multi-institutional basket trials that are linked to the MASTER platform and that all assume that the presence of a specific molecular marker predicts response to a targeted treatment independent of tumor histology. And a specific example is shown here. This is a randomized phase 2 trial in which we are evaluating the efficacy of combination treatment with a PARP inhibitor, olaparib, which I mentioned before, and the conventional cytotoxic drug, trabectedin, in patients with genetic features of impaired DNA repair via homologous recombination. And this is an ongoing trial, so there are no final results. But I can tell you that we have randomized 72 patients. These patients represent more than 10 entities, including soft tissue sarcomas and also chordoma cases. And the first interim analysis has demonstrated non-inferiority of the investigational treatment and found that a substantial proportion of the crossover population that failed standard treatment appears to benefit from experimental treatment with olaparib and trabectedin. This chart provides an overview of another other molecularly stratified clinical trials that are currently linked to specific intervention baskets of our diagnostic platform. Our dream, of course, is that we can link each and every intervention basket to an interventional clinical trial. And I wanted to mention one specific protocol that we are excited about at the moment. So last week we opened CRAFT. CRAFT is another phase 2 trial that comprises 6 different molecularly defined treatment arms. And the unique feature of this protocol is that we will be investigating combination treatments. So combinations of targeted agents that we assign according to the patient's genotype and a PD-L1 antagonist, so immunotherapy in most treatment arms. And the idea here is to counteract intrinsic or acquired resistance to therapy. So this is just started, first patient is in, but this is something we are excited about. And with the following 2 slides, I just wanted to address that an essential task of a research-oriented precision oncology program like MASTER that is rooted in the DKFZ is, of course, exploring new levels of patient characterization. And in this regard, we are trying to take 2 approaches. The first one is to attract new clinically actionable information from the data that already exists, so that are already being collected using bioinformatics analyses. And this is mainly the work of Daniel Hübschmann and his team, Benedikt Broers and colleagues, and many others in the bioinformatics community here in Heidelberg. And we are currently focusing from a clinical perspective on complex biomarkers that will hopefully allow us to predict response to different immunotherapy modalities. We are looking at the estimation of signaling pathway activity using RNA data. The identification of alternative telomere lengthening is of interest. And we are also interested in pharmacogenomic risk prediction. And this is something we are doing together with Matthias Schwab and his team at the Institute for Clinical Pharmacology in Stuttgart. And the second approach, of course, is to generate new levels of data using methods beyond genomics and transcriptomics. One specific example is the MASTER Pro study. This is a DKTK initiative led by Wilko Weichert and Bernhard Küster at the Technical University in Munich. This is a pilot project that aims to establish a workflow that allows us to, by mass spectrometry, measure the expression of approximately 8,000 proteins and 15,000 post-translational modifications in parallel to our genomic and transcriptomic analyses. And in the intermediate term, or maybe the short term, we're trying to integrate the results into clinical decision-making. And this level of data is new territory for us, and this is beyond routine clinical care, and brings me to another and last practical aspect that I would like to mention, and that is the importance of interdisciplinary molecular tumor boards for the evaluation and integrated consideration of all clinical and molecular data on a given patient. Mike Berger mentioned that they never established an MTB at MSKCC. I think for our center that has worked really well, these interdisciplinary conferences, and this is just our very practical setup in Heidelberg. So at NCT Heidelberg, 3 molecular tumor boards are held per week on Wednesdays and Fridays. Comprehensive molecular profiling results have been discussed for several years as part of specialized programs such as the MASTER program, but also the CATCH and cognition studies that focus on patients with advanced stage or potentially curable breast cancer. And last year, we also established in close collaboration with our colleagues in Dresden and our colleagues at the Institute of Pathology a molecular tumor board to discuss panel sequencing results in patients with solid tumors. And last but not least,
sorry to interrupt, we also have to have many questions already lining up for you so that we can answer them.
My very last slide is the only data sharing slide that I have, so maybe I should present it very briefly. Last but not least, I just wanted to highlight that sharing data out of the MASTER program has been relatively slow and inefficient so far, as this is a process where our coordinators, who also created this slide, and thanks to Annika Baude for the last minute help here, have to go through a very complex and tedious workflow with many steps and bilateral interactions within DKFZ and with external partners. And this issue is gradually becoming a relevant bottleneck for our work, with now more than 70 spin-off projects that came out of MASTER. This is my summary slide. I'll skip that. But with my second to last slide, I just wanted to acknowledge Pass Pro Toto, everybody within the German Cancer Consortium who helped establish this program. As you can imagine, there are hundreds of people, especially in Heidelberg and Dresden, who have contributed. Thanks to all of them. And thank you for listening, and thanks again for the invitation.